goal of my research is to restore motor and sensory function after nervous system damage or disease. To do this, we'll record from or stimulate the peripheral nerves, which are like the hot wires from the brain to the rest of the body. So for example, suppose a person has spinal cord injury. In that case, the nerves still work and they're still connected to the muscles, but they're no longer receiving commands from the brain. Consequently, if we could stimulate and activate the motor fibers in the nerves the way that the brain normally does, then the muscles would move the way that they normally do, and thereby we could restore walking or restore hand function. Or to pick another example, suppose a person were missing part of their limb. In that case, we could use a recording device to wiretap into the nerves, capture the commands that are coming down the nerves that the brain would be sending normally to the muscles, which are no longer there, and then use those motor commands to move an artificial limb. Or similarly, we could stimulate the sensory fibers that go back up towards the brain and thereby communicate sensory information about touch or about movement or even the sense of self. And to do this, we are investigating the use of a device called the Utah Slanted Electrode Array, invented here at the University of Utah by my colleague Richard Norman. That device is shown here at this end of the wire bundle. Too small to see, there are 100 individual hair-thin electrodes in this device. And what these multiple electrodes allow us to do is to talk to or listen to nerve fibers very selectively and very precisely. And in so doing, we can communicate with the nervous system in a manner that begins to approach that of biological normal physiology. And so our hope is that by communicating with the nervous system in a biologically realistic manner, we'll also be able to restore function in a biologically realistic manner as well.